So hello everybody, I'm Dean Whiting here from Pathology Technology Australia, and today I have a very special guest, Mr. Wayne Dimech. Wayne is the Executive Manager for Scientific and Business Relationships at NRL, and not the National Rugby League, but the National Reference Laboratory for the Quality Division. So Wayne and I have known each other for a very long time. Wayne has been in and about laboratory services and the NRL for decades, and I'll let Wayne explain in a minute. But of most importance, I think you should I should point out Wayne is a fellow of the Australian Institute of Medical Scientists and also a scientific fellow at the Royal Australian College of Pathologists. So Wayne, welcome. Is there anything else we should know about you at this stage?
No, not really. Just that I've been in the business of improving quality for test kits for many decades now.
That's great. Look, I thought it was really opportune to talk to you at the moment because there is a fair bit of discussion around at the moment on the on serology testing for COVID-19, and I think that's a topic that I think there's a bit of misinformation around. There's good information also. And I think as we talk about changing from the detection phase into the detection and management phase of the COVID pandemic, it's worth understanding what tests are available for serology testing for the antibodies and antigens. So I wonder, Wayne, just in the first instance, can you just go through, maybe list out what sort of testing would we be thinking about? What would be available for serology testing in COVID-19?
Sure. Well, as you probably are aware, the first response for serology testing for COVID-19 were mainly rapid tests. They came out very quickly, and many of them, most of them purporting to detect both IgG and IgM. More recently, though, the larger manufacturers have started coming out with test kits for the automated laboratory platforms, which are commonly used in serological testing in developed countries. What we do know, though, is that for IVDs, for infectious diseases in particular, other than COVID, regulators require the IVD manufacturer to show evidence of the quality of the performance of those assays, that they need to provide the regulator comprehensive data on the performance of those assays, and it is only until the regulator is satisfied that those assays can be sold into the country. But under COVID-19 testing, the regulators have removed that requirement. They've have an emergency use listing for serology and for NAT testing, which means that the manufacturers don't have to provide the evidence that we usually are associated with test kits that are on the market. And so now what is happening is we have, in about 5 months, over 600 test kits for COVID-19, and we actually know very little about the quality of those test kits.
Yes, and I read the literature and the newspapers around this topic greatly, as you might imagine, and I do see a lot of fairly bad press about some of the antibody tests, and unfortunately many of the tests have not lived up to the manufacturer's claims and have not turned out to be at all useful, which has led, I guess, to a World Health Organization, to RCPA, and a couple of state healths in Australia, Western Australia and South Australia, namely, have strongly recommended against the use of some of these lateral flow assays. But as you say, there are a range of laboratory-based assays available as well. And your service has for years validated the performance of many of these assays, the hepatitis, HIV classically. Yes. How would you go about evaluating the performance of these serology tests, and what would you look out for, and why would you look out for these things?
Sure. So there are actually some really well-established criteria for evaluating assays when a manufacturer does provide that information to the regulator. They need to not just look at the sensitivity and specificity, as important as that is, but they look at other factors. They look at reproducibility, repeatability. They look at limits of detection, lot-to-lot variation, really critically, they look at the analytical sensitivity of the assays using seroconversion panels and other criteria such as interfering factors and cross-reacting antibodies. You need to look at all of those elements to do a comprehensive evaluation. If you go straight into a laboratory verification, what laboratories are most used to, doing small numbers of tests, that actually is less meaningful if you don't have that solid foundation of evaluations that we usually require from a regulatory purpose. And so we need to accumulate that evidence. And to be honest, we haven't really seen any of that sort of information coming out into the public domain yet. So NRL is using its expertise. We have well-established processes to do this. And we are acquiring suitable samples to do this evaluation. And over time, we hope to be able to present that information, because without that, most of the other understanding of the usefulness of serology testing is going to be compromised.
Yes. No, I think that's right. And I think there's a little bit of a hump to get over now with some of the bad press that the assays have been acquiring up until now, or at least the concept of serology testing in pandemics such as COVID. We've acquired somewhat of a bad name. So I think something which from an independent authority, something which evaluates the performance is going to be important. And I think your service will offer that. Immunity in general, then, I mean, it's important, but around serology, around the COVID-19 serology, how do you think that testing the antibodies is going to help us maybe with, determining the immune status of individuals and of the population?
Sure. So the first thing is, in general, serology has a really well-established situation within the testing of a patient and in their cascade of care. So we generally associate the presence of antibodies to indicate an immune status or a past infection at least. We associate the absence of antibodies as that person being naive and susceptible to disease. We also use different classes of antibodies to determine whether a person's been recently infected, for example, using IgM antibodies. And obviously, serology has a place in seroprevalence studies in understanding what the herd immunity is. But for COVID, we're again in a slightly different situation. It's a really bit early. We need to determine whether everybody produces an antibody response. We really don't know this. You and I shared some information this morning about, in the New York Times. Yeah, we don't know whether a person is, every person who gets infected gets an immune response. We don't know how sustainable that immune response is. We don't know whether individuals who have asymptomatic or a mild disease has the same immune response as someone who has a florid disease. So until we understand the immune response better, we are really not going to understand exactly how COVID serology testing is going to fit in. But we're not going to understand that immune response until we have really good tools. We have to understand the tools, the serological assays, before we can understand how that immune response is affecting our understanding of the disease.
Yeah. I think that's absolutely right. I mean, and then it's almost a chicken and egg situation here. What comes first? Do we test first and then figure out what the immunity is? Do we figure out what the immunity is and then test? In this case, probably the cascade needs to be, let's make sure the assays do what we think they do, then start measuring a wide enough group of the population to start seeing. And the New York study that you referred to a minute ago it's not yet peer reviewed, obviously. So there's a lot of water to go under the bridge, but that appeared to show that even asymptomatic people, regardless of your age, sex, or the severity of your challenge by COVID-19, you develop the antibodies. Now, there's there's a lot unknown about that study. I mean, how did they collect the sample? I mean, who participated? Is it a truly random set, or is it somehow, a biased set of samples? But nevertheless, it's from a very good school and a very reputable research group, and so it's quite encouraging, I think, what they've shown. So one thing is validating the performance of the test now, but on into the future, what's the important role of ongoing validation of these assays? Because we know that these are biological systems. They're sometimes challenging to manufacture. Raw materials can change. The antibodies in nature might change. What do you see as the role of ongoing surveillance of the performance of such tests?
Absolutely. So when you do an evaluation, it's an evaluation of a test kit at a point in time. It's usually done in ideal circumstances. You're actually wanting to evaluate the test kit rather than the environment that it's being used. Once that test kit is into the laboratory in routine use, first of all, we're in a unusual situation. The manufacturers are ramping up production. We're starting from zero up to trying to supply the whole world all at one time. There is competition for components. We see the competition around PPE and other swabs and viral transport media. The same thing is happening for monoclonals, for reagents, for buffers in the test system. So we can assume that the supply chains for manufacturers are potentially stretched. It's only been 4 months since that has started. So we expect that the manufacturing processes and the contingencies for other materials is somewhat immature. And we also know that there are many manufacturers out there who have never been manufacturers of infectious diseases, IVDs. So we're in a situation where we need to monitor the performance of these test kits even more than normal. And even in a normal situation, we know that there are time-to-time failures in production. We just published, just a couple of months ago, a issue that we found with a commonly used hepatitis C kit from a very reputable manufacturer, where they changed out a non-analytical component, and it really caused problems. So, and we also know that there's batch-to-batch variation in rapid tests. That's a really common thing that happens, and that affects performance. So we generally monitor the performance of assays through two things. One is external quality assessment schemes, and the other one is a run control. Quality assessment schemes are essentially mandatory in most developed countries, and so we expect people to be participating in those schemes, and that's a scheme that NRL will be providing as well, both for serology and for nucleic acid testing. But I think critical to me is being involved in a run control program, a program where you're testing the same sample every day and monitoring that sample result in a validated process. And that's something that NRL has provided for infectious diseases testing laboratories all around the world for more than a decade now.
Yeah, I think that's critical, and I mean we've had a lot to do with each other, I guess, over the years in some of the NRL serology type quality assurance programs that you have established. Those are quite, your services have been used in many countries around the region, haven't they? Give us some idea of how far your service reaches.
Well, our EQA programs go to around 70 countries around the world. We go all across Europe, all through Africa, through the Caribbean, and obviously Asia. So we have a very wide footprint.
Yeah, and it's a very valuable service, I know. I mean, the kinds of services you offer are pretty unique. Not pretty unique, you can't qualify the word unique, but there are very few people offering the same kinds of services that you offer. I know as part of a previous organisation we were using Paul Ehrlich Centre, and yours would be at a par with that, wouldn't it? The kind of services being offered.
Yeah, absolutely. There are many organizations that do same things as us, but there's no one organization in the world that does test kit evaluations, that do quality assurance, external quality assessment schemes, that does QC programs, and that does training for developing countries. I'm pretty sure that I can say categorically we're the only organization in the world that does all of those things. But the thing is, they're all intertwined, they're all inseparable, they all build on each other.
Yeah, very good. Okay, Wayne, I think that's been terrific. Our time is up, unfortunately, but yeah, I really appreciate the opportunity to chat about this with you. Look, if anybody has any further questions, they can get in touch with you at NRL, I'm sure,
Absolutely.
or get in touch with Pathology Technology Australia on our website. So I'm Dean Whiting, and thanks very much to Wayne Dimech, and bye for now.
Thank you, Dean. Bye.